Hello. So my experimental design was looking at the effects of different base layers on kidney bean growth. So my hypothesis was if kidney beans are placed in cotton, then they will grow the most. And in this, we can see that the independent variable is types of base layers for the beans to grow on. I chose 5 types of base layers, or my treatment groups, which were paper towel, cheesecloth, sponge, cotton, and fabric. I took a little piece of all of these materials and I put it in a plastic bag. And in these materials, I had exactly 5 beans. So each treatment got around 5 beans each. And to do my experiment, at the end, I kind of, like for example, for cotton, I took the 5 beans and I measured its length. And I took a mean of it to get my result. And the result, as you can see, would be the dependent variable, which is the growth or the height of the kidney beans. And I took the mean to see which material showed the most growth in the kidney beans. In this experiment, my constants were that all the material, like all the treatments, got the same amount of light, which was yellow light, every single day, and their data was recorded at the same time. And I used somewhat a same method to kind of give them water. So before I put in my material in my Ziploc bag, I dipped all my material in water and gently squeezed it, and then I put it in my Ziploc bag and put my beans in to let the experiment start. And in this experiment, my control group is paper towel, which is usually what people use to experiment or grow beans in. For my video, I don't have the actual beans that I could show because I threw them away. However, I could describe what my beans looked like. Especially the biggest changes were seen in cotton, in which I was able to see an actual growth of leaves. And I think mostly all 5 of my beans in the cotton treatment had leaves or some sort of a little plant growing out. While in fabric treatment, I got to see some very long roots that were embedded inside the fabric. And for my cheesecloth and my paper towel, I was able to see some really small, like not very small, but medium-sized roots that were embedded inside the actual material. But there was not a lot of plants or any leaves coming out. However, my sponge, which was the last one, didn't have a lot of growth. It had some stem coming out, but it did not have a big growth in terms of the others, that like cotton and all the other ones, in which it looked like if I kept them more in my treatment or kept the treatment going, there would be plants coming out of it. No, sorry, not plants, but like some tiny leaves and other things coming out. But for my sponge, no big difference was being seen. Only a little bit of a stem was being grown out. For my last part of the video, which is the analysis of my results, I can talk and say that my hypothesis was proven true, that cotton did show the most growth. As even looking in the graph, I could see that compared to all the other ones, cotton had the most growth. But when we look at the SEM, we get to see that the cotton and fabric kind of overlap each other. Their SEM overlap each other, so we could say that they had almost the most growth. And then all the other experiments like paper towel, cheesecloth, and fabric, they kind of overlapped each other, showing that they had a lot of similar results. So their growth, their lengths were very similar. And sponge was the only one I would say that was really at the bottom and did not grow as much. So it was probably one of the least amount of mean values of bean growth that was being seen. So from my results, I could say that mostly all of them, looking at the SEM, we could say they kind of overlap. So they had mostly the same values. But the only one that really stood out was cotton, which showed the most spike. But looking at the SEM, like I said, it is overlapping with fabric. So we could say that fabric and cotton are one of the ones that gave out the most bean growth. And all the other ones were kind of in the middle, overlapping with each other, like overlapping the SEM with each other. And sponge was probably at the bottom with the least amount of growth being seen. And that was the analysis for my results. Thank you.